This is the first lecture in which we will talk about cells, and we will talk about 4 topics related to them. First, we will cover microscopy, which is an important technique in studying cells. Second, we will talk about cell theory, which has 3 components and talks about the importance of cells to living things. Third, we will talk about cell size and why large cells do not work as well as small cells. Finally, we will talk about one group of organisms that are almost all single cellular, and these are the prokaryotes. We'll talk about 2 main groups and some characteristics of each group. And so here's that list of topics again: microscopy, cell theory, why cells are small, and prokaryotes. We'll start by talking about microscopy. In microscopy, our goal will be to magnify as well as resolve structures. Magnify simply means making larger. So for example, I can take this small image of a pollen grain and just stretch it and make it 5 or 10 times bigger. And that is useful, but that is not adequate. The other thing that we need to accomplish is increasing the resolution. So resolution is the ability to distinguish nearby structures as being separate. In a low resolution image, such as this one to the right, we cannot individually distinguish nearby bumps on the surface of the pollen grain. They blend together and it sort of looks like a smooth surface. In contrast, for that same image shown at the left, this is a higher resolution image because nearby bumps on the surface are individually distinguishable. And so we can see some details here in the high resolution image we could not see in the low resolution image. With microscopy, our goal is to have highly resolved images. So images where we can see very small objects. It turns out that the wavelength of light or the wavelength of electrons that we use in order to visualize our object is going to be important to the size of the object we can detect. And here's why that is. Waves miss objects. They simply fail to hit them if the object is less than about half of the wavelength. As an example of this, look at this top wave. This has a long wavelength. In other words, from one peak to the next peak is a long distance. And the object we're trying to view, this rubber ducky, is small relative to this wavelength. In this example, this wave could pass entirely around the rubber ducky without ever hitting it, even if the duck was in line with that wave. As a result, this wave would be ineffective at visualizing this duck. In contrast, let's imagine we had a shorter wavelength and we passed a beam of this wave towards this duck. In this case, most of the waves would in fact encounter the duck, and we could use what happens as a result when those waves hit the duck in order to visualize the object that we're looking at here. So the general lesson from this is that waves that are long are less effective at detecting small objects than short waves, and shorter waves are better at detecting small objects. An upshot of this is that when we have a choice in wavelengths, we will often use shorter ones when we are trying to image very small objects. Let's look at this with regard to visible light, which is what we use with compound light microscopes compared to electron microscopes. Visible light has wavelengths that are about 400 nanometers or longer. And so half of that is about 200 nanometers. So according to this calculation, we can only use visible light to image objects that are about 200 nanometers long or longer. And here's just another example of pollen grains. These are what lily pollen grains look like when visualized with a light microscope. We can see some features. We can see that there are some bumps on the surface, for example, and we can see the general shape. However, if we instead use an electron microscope, this is basically a microscope that shoots beams of electrons at an object, and it can do it in one of two different ways. The example here is from a scanning electron microscope. That means it's shooting beams at the surface, and those beams are bouncing off. The wavelengths of electrons used in electron microscopes are somewhere on the order of 1,000 times shorter than the wavelengths of visible light. This means that we can resolve objects that are much smaller. So we can resolve objects as small as about 0.05 nanometers with an electron microscope compared to about 200 nanometers with a light microscope. That is a marked improvement. And what we see here is a scanning electron microscope image of a lily pollen grain, the same kind of pollen grain as above. We see the same general shape, but you can also see a lot more details of what the surface of this pollen grain actually looks like. You can see each of these little plates that form the surface, which you could not see up here. Another way of saying this is that the scanning electron microscope image is much better resolved than the light microscope image. So with this in mind, we can compare and contrast the advantages of compound light microscopes, such as you'll use in laboratory, with electron microscopes. Compound light microscopes are good because it is possible to view living specimen, at least in some cases. One can view color because there are all of the wavelengths of light that can pass through. They are also relatively cheap, and it is relatively quick to prepare slides. In contrast, for electron microscopes, the big advantage is that because the wavelengths are much shorter, there's much higher resolving power. There are two types of electron microscopes. So before we go to the types, the problems with electron microscopes is that they can be very expensive to both to buy and to maintain, and the process of preparing specimens to view is relatively more complex. There are two types of electron microscopes: transmission electron microscopes and scanning electron microscopes. With transition electron microscopes, which you will often see referred to as TEMs, the idea is that a beam of electrons passes through the object that is being viewed, much like light passes through the object with a compound light microscope. In contrast with a scanning electron microscope, which you'll see abbreviated as SEM, what happens is that the beam of electrons is reflected off the surface of the object, and it's the bouncing of those electrons off the surface that creates the image. So if you want to know what the outside looks like, you use a scanning electron microscope. If you want to know what the inside looks like, one would use a transmission electron microscope. If we look back at this image of a pollen grain, we're seeing the surface. So this must have been an SEM image. Before the invention of microscopes, we did not have strong empirical evidence that all life was cellular. But as we looked at living things more and more after the invention of microscopes, we came to realize that this is true. And with this, ideas of cell theory were developed. Cell theory has three main ideas that are now commonly accepted. The first idea is that all life is composed of one or more cells. So some living things are made of only one cell, and these are called unicellular, uni meaning one. In contrast, other living things, such as us, are more than one cell or multicellular. And so here are just a couple of examples. At the far left is a bacteria, and almost all bacteria are unicellular. This one is Yersinia pestis, and it happens to be the bacteria that causes plague. We also have eukaryotes, which we'll learn very soon are more complex cell types with internal membranes. But even for eukaryotes, there can be unicellular organisms. And here's an example. This is Giardia, the organism, a protist, which causes diarrhea and life-threatening conditions. and this is a disease that still kills many people, especially in developing portions of the world. Finally, over here at rate, I already said that we as humans are, of course, multicellular, but any living thing that you can see easily with the naked eye is probably also multicellular. So just as one random example, this is a red algae, which would grow in a marine environment, and it is a multicellular organism, in this case attached to the surface of a rock. So to summarize point 1, cell theory says that all life is cellular. Cell theory point 2 is that cells are the smallest unit of life. Of course, cells are made up of smaller organelles and macromolecules, but the parts of cells themselves are never self-sufficient and cannot survive on their own. As a result, we wouldn't refer to particular organelles of a cell in themselves as being living things, even though the cell could be referred to as a living thing. So cells are the smallest unit of life. The third aspect of cell theory is that all cells arise from pre-existing cells. What this means, the implications are actually fairly striking. What we're looking at here is the tree of life, showing several of the major branches. And over here are the eukaryotes. These are the more complex cell types, and it includes things like animals, fungi, and plants, as well as many other groups that we lump together under the term protist. But since every cell arose from a pre-existing cell, that means within your body, of course, that's true. But at some point, your cells arose from your mother's cells, and your mother's cells arose from her mother's cells, and we can trace that back in time, and eventually we can trace back all animal cells to one initial animal cell. On the diagram, that would be somewhere back here. And we could say the same thing for fungi. All fungal cells arose from pre-existing fungal cells until we got back to this point where there was an ancestral cell that gave rise to one offspring cell that eventually evolved into animal cells and another offspring cell that evolved into fungal cells. And going back just a hair further, there would have been an interaction or a point where a cell gave rise to a offspring that became all plant cells and gave rise to another offspring that became the parent of all fungal cells and animal cells. And we could trace that back further and further and further until this point on the tree, where there was a cell that gave rise both to all Archaea, and this is a group of prokaryotes we'll talk about momentarily, and its other offspring gave rise to all eukaryotic cells. And then going back to here, we would have had a split between a cell that gave rise to all bacterial cells with one offspring and all other cells, including both Archaea and eukaryotes, with its other offspring. And then we can trace it even back further to this point on the diagram, which would be the first cell to have formed. Now, there's a question: if all cells came from pre-existing cells, then where did the first cell come from? This is sort of like the chicken and egg question. There must have been one first cell. So that first cell would be the one exception to this point in cell theory, where a cell actually must have arisen without a pre-existing cell. We know something about the conditions on Earth that would have allowed a lot of the molecules necessary for life to have existed. But how they all came together to form a cell is something that still has a lot of uncertainty surrounding it. Or it's possible that instead an initial cell came to Earth from space, but that sort of just sets the question back even further. That cell at some point must have arisen from non-living material, whether it occurred on Earth or somewhere else in the universe. So those are the three components then of cell theory: that life is composed of one or more cells, cells are the smallest unit of life, and all cells arise from pre-existing cells except the first one. We will move on here and talk about why cells are small. And the key idea is that cell size is determined by a combination of the cell's surface area and the cell's volume. To think about this, we're going to do some abstraction. Cells are not technically spheres, but it's nice if we just imagine a cell as being a sphere, and then we can think about what would happen to that sphere, and we can say those same rules probably apply to similarly shaped cells. So if this sphere was actually a cell, it would need to do some things to stay alive. First, it would need to import nutrients from outside. If it was unicellular, maybe this would be floating in the ocean, it would need to import nutrients from that ocean water. But even if it was multicellular, so a cell in a muscle cell, let's say, then this cell would still need to import glucose, it would need to import oxygen, and it would need to import other nutrients to survive from the fluid in your body. Then the second thing the cell would need to do is export waste. So as the cell is doing respiration, for example, it would be making carbon dioxide, which would need to leave the cell. And both of these processes, importing and exporting, happen across the surface area of the cell. So the more surface area, the more the ability to import nutrients and to export waste. Now let's think about the volume. If you think about the cell as a factory, then the larger the size of the factory, or the larger the volume of the cell, then the more materials are needed. So a small factory only needs a small amount of inputs, a large factory needs more. Similarly, a small cell only needs a small amount of nutrients, and it only has a small amount of waste to export. But the larger the cell is, I can make this larger, then the more nutrients it needs to bring in, and the more waste it needs to export. So if the surface area increased at the same rate as the volume, this wouldn't be a problem. As the cell got bigger, it would need more stuff, but it would also have more surface area across which to import and export. It turns out that that's not the case. It is true that larger cells have larger surface area, and they also have larger volume compared to smaller cells. However, this is the important idea. The volume of a cell increases at a higher rate than its surface area. This means that as a cell becomes larger and larger, the ratio of the surface area to volume actually decreases. Now, I'm going to say this one more time because students often get it confused. The surface area is getting larger. Of course, a big ball has more surface area than a small ball. So the surface area does not get smaller. As a ball gets larger, it gets larger. However, the volume gets larger at an even faster rate than the surface area. So the amount of surface area relative to volume is decreasing as the cell gets larger. If we think about our factory example, what we're saying is that a larger factory can have more loading docks than a small factory, and a large factory can have more equipment than a small factory. But as a factory gets larger, if the factory is shaped like a sphere, then the larger the factory gets, the fewer loading docks there are per piece of equipment inside that factory. So it gets harder and harder to get all of the supplies necessary into that factory as it gets larger. Of course, we wouldn't design factories as a sphere, and not all of our cells are spherical. But this still gives us a general idea of why it's hard for an object to get larger, or at least for a living cell to get larger, without running out of surface area for import. So because of this problem, the solution is that as a cell gets bigger and bigger, it needs to divide. And by dividing, it becomes two smaller cells, and that in effect increases its surface area relative to its volume, and it helps restore its ability to import and export the amount of waste necessary for that cell. So this is why, in most cases, Even large organisms are made of many small cells instead of a few big cells. And there are some exceptions to this, but we're going to worry for now about what normally happens, which is that cells tend to stay small. We have made good progress so far. We talked about microscopy with light microscopes as well as scanning and transmission electron microscopes. We've talked about cell theory and its three components, and we've talked about why surface area to volume ratio means that small cells tend to be favored even in large organisms. We're now going to move on and talk about a group of organisms, most of which are single cellular, and these are the prokaryotes. Prokaryotes are organisms that have generally small cells and less complex cells than the other group we'll talk about later, which are the eukaryotes. Prokaryotes, as we saw on the tree of life, which we'll look at again, were the first cell types to evolve, and they later gave rise to a lineage that evolved into the eukaryotes. Here is that tree of life we were looking at with the first cell indicated down here. And the first two groups to diverge, so this group here is the bacteria, which we'll also call the Eubacteria. Just add an EU in front of the word bacteria, and we can use those interchangeably. So that lineage arose, and then the second lineage arose. The second lineage had a group that branched off early, and this group is called the Archaea. These two groups together, the Archaea and the bacteria, comprise the prokaryotes. And as I said, we can add U, if I can write that with my mouse, in front of bacteria to call these Eubacteria. And so these are the two main groups of prokaryotes. And we'll talk a little bit about each of the groups. We will start by talking about characteristics of the Eubacteria, but these are going to be similar to what we see for Archaea. Eubacteria are small, they are typically single cellular, and they have simple structures. By this we mean in part that they have no membrane-bound organelles. We'll clarify what we mean by membrane-bound organelles momentarily. And Eubacteria are the most common group of prokaryotes. If you were going to be made sick by a bacteria, it would probably be a Eubacteria. Most of the Eubacteria in your digestive tract are Eubacteria, and most of the bacteria in the environment also belong to this group. And I'm going to see now if I can get this to play. This is just sort of a neat video showing time-lapse of a single bacterium, this white spot in the video, dividing over time. And we'll see that as the number of minutes go by, the number of bacteria in the colony that form. Each time a new bacteria is formed, it stays in the same place. So we end up seeing a colony. It looks sort of like a multicellular organism, but each of the bacteria is operating on its own. It just happens to be next to another bacterium. So let's see if we can get this to play. Here we go. So that first one divided into 2. Now each of those divided into 2, and the 2 became 4, the 4 became 8. And very quickly, we're up to about 4 hours. You can see 6 hours, we have many, many times more bacteria than we started with. So this is showing a phenomenon called exponential growth. And this is why it's important, for example, to keep food refrigerated so we don't have bacteria that quickly reproduce over and over into forming large numbers. Before we move on, let's look at a couple of examples of Eubacteria which are in this group of prokaryotes. So one example is a gut bacterium, Escherichia coli, which is pictured here. This is actually several of them that are sort of stuck together. And this bacteria is present in the guts of all humans, and most forms of this bacteria are harmless and probably desirable to have in our guts. However, there are some strains or genetic lineages which cause illness in humans and can even cause death. And so when there's a recall because of E. coli contamination, what really we're doing is trying to remove from the food stream food that's been contaminated by some of those forms that do cause sickness. So that's E. coli or Escherichia coli. Another example is a nitrogen-fixing bacteria called rhizobia. And rhizobia can live in the soil, but they can also become trapped and engulfed in root cells where they have a symbiosis with the plant. Either way, they are going to make their living in part by converting atmospheric nitrogen, or N2, into ammonium, or NH4. Ammonium is a form that can be used to do things like make amino acids and can be used by the plant as a nitrogen source. N2 cannot be used by plants as a nitrogen source. And so rhizobia play an important role in making nitrogen compounds available to plants. What we see in this image is a cross-section of a root cell, and these dark spots are enlarged bacteria, enlarged rhizobia bacteria inhabiting parts of the root. So we've talked a bit about the Eubacteria. We'll next talk about the Archaea, which is this group here. Eukarya, or rather Archaea, and Eubacteria are similar in size and simplicity. And you can see that, at least in this image. This bacteria here, labeled Si, is a Eubacterium, and these over here, labeled delta H, are Archaea, and so they look very much alike. Despite this, if you actually look at DNA sequences, or if you look at the details of the structure of the ribosomes, we find that there are more similarities between Archaea and eukaryotes than there are between Archaea and prokaryotes. This tells us that Archaea and eukaryotes share a more recent common ancestor with each other than either does with Eubacteria. I'll say that one more time, then we'll look at the diagram. Archaea and eukaryotes share a more recent common ancestor with each other than either does with Eubacteria. Let's go back here to our diagram of relationships. What we're saying is that Archaea and eukaryotes converge to this common ancestor right here, and that common ancestor occurred between them before the common ancestor of either of those groups and the Eubacteria shown in blue. That common ancestor was earlier in time, way back here. So Archaea are eukaryotes' closest relatives, and eukaryotes are Archaea's closest relatives. Archaea are an interesting group. The groups that we talk about most are the ones that live in unusual environments. For example, there are Archaea that live in salt lakes, like the Great Salt Lake, as well as in hot sulfur springs that would be way too hot for you to put your hand in without scalding. And there are also Archaea that live in either very acidic environments, so very low on the pH scale, or very alkaline environments, very high on the pH scale. And so these are unusual. However, there are also many Archaea that live in entirely normal environments. So there are Archaea that live in marshes and in soil and in the oceans and even in the guts of humans, as well as ruminants like cows and termites and probably most other animals. And they in fact play an important role in breaking down organic material. One role that a particular group of Archaea provides is breaking down organic matter and forming methane, or CH4. And so this is a complete breakdown of carbon products that can happen in the absence of oxygen. And it can happen in humans. It's especially important in the guts of animals like ruminants like cows, and also in the guts of termites that are eating wood. So here's a termite over here that would have archaea in its guts. To summarize the groups we've talked about, the two main groups of life we could consider to be prokaryotes and eukaryotes. The prokaryotes would include the Eubacteria, also just called bacteria. You can see it either way, and the Archaea, which are sometimes called archaeabacteria. I'll put both of the names here so you're not confused. Then the other main group are the more complex organisms we'll be talking about later, and these are the eukaryotes. These do have membrane-bound organelles. They do have a nucleus, and they are more likely to be multicellular. And this will include groups like the protists, which have many single-cellular and many multicellular members, and then the fungi, the animals, and the plants, as well as some other groups. We're going to focus for the remainder of this lecture, we're going to stay focused on the prokaryotes. And we're going to talk a little bit about their anatomy. We've said that they're simple. They are non-compartmentalized. What this means is they have no membrane-bound organelles. Membrane-bound is in italics and bold because it's important. Prokaryotes do have organelles. The one kind of organelle that they have that we'll talk about are ribosomes. And so remember, ribosomes are used to make proteins, and ribosomes are considered to be organelles. They simply do not have a membrane around them, so they are not membrane-bound organelles. So this is the important kind of organelle that you should know prokaryotes do have. Remember that the nucleus is a membrane-bound organelle within the cell. Because prokaryotes do not have membrane-bound organelles, they by definition cannot have a nucleus. They do, however, have DNA, and you can see that as the red swirls in the middle of this bacterium. And so what we call the area that has the DNA is the nucleoid region. To go over some basic structures in prokaryotes, let's think about some structures in prokaryotes as well as all other cells, so also things that are in eukaryotes. They have a cell membrane, which is this lipid or phospholipid bilayer that we'll spend a lot of time talking about soon around the outside of the cell. They have cytoplasm, which is the semi-liquid matrix inside the cell within which all organelles and all substances are located. They have ribosomes, which are the organelles used to make proteins. And of course, they have DNA. So those are in all prokaryotes as well as all eukaryotes. Prokaryotes may have a flagellum, which is this tail-like structure that can be used for things like propulsion. Now, it's worth noting that eukaryotes may also have flagella. Many eukaryote cells do not, but some do. For example, sperm cells, and also cells in sponges, and also some plant cells. Some plant reproductive cells have flagella. So this is not a structure that is unique to prokaryotes, even though it is found in some. Another structure that is found in prokaryotes is a cell wall. In bacteria, or in eubacteria, this is typically made of a substance called peptidoglycan, which is a big word, but you should recognize both of the roots. So peptido is basically referring to peptide bonds. And peptide bonds should remind you of amino acids connected together. So really the peptido part of this word is saying that this substance includes amino acid chains. The second part of the word is glycan, and glycan is a term that refers to polysaccharide chains. And you already know about polysaccharide chains, for example, in starch or cellulose. So peptidoglycan has both amino acid chains and polysaccharide chains. You can see that in this diagram. Basically, what we're looking at is the structure of this substance. And there are polysaccharide chains shown by these balls connected by little sticks. And then they are cross-linked to each other by these shorter chains. These shorter chains are short chains of amino acids or short polypeptides connected to one another. And that mesh together forms this substance forms the substance that is the cell wall in bacteria. Archaea also has a cell wall. It is not made of peptidoglycan, but it is made of some other materials, which we will not worry about. Then finally, outside of the cell walls, we've got plasma membrane closest to the cytoplasm, and then cell wall in the middle, and then the furthest out here is a layer that's often present called the capsule. The capsule is a jelly-like substance, and it aids in the bacteria attaching to surfaces, and it helps it form films. So this isn't always present, but it is frequently present. This table summarizes much of the material we've just talked about. So it contrasts prokaryotes with the bacteria or eubacteria and archaea or archaebacteria, and the eukaryotes, which include protists, fungi, plants, and animals. The location of the DNA is loose in the cytoplasm in prokaryotes, in the area that we call the nucleoid region, while in eukaryotes it's enclosed in a membrane-bound organelle called the nucleus. Organelles are present in prokaryotes, and ribosomes are the ones that we will be concerned with. And they are also present in eukaryotes. There's a greater variety, in fact, in eukaryotes. Are there membrane-bound organelles? In prokaryotes, no. In eukaryotes, yes. And how big are the cells? Well, there's variation within each group, but on average, prokaryotes are typically much smaller than eukaryotic cells. To summarize overall today's lecture, first, you should know the three main lineages of living things that we've discussed and some basic characteristics of each, especially for the two prokaryotic lineages. You should know characteristics of eubacteria and archaea, and you should know general characteristics of prokaryotic cells. Also, you should be able to explain why cells stay small and divide rather than getting larger and larger. And when you do this, you should be able to explain it based on surface area to volume ratio.